to whichever one works for you. So let's get started. So please know, have your outline with you. Have a highlighter and a pen. So you can go through this and we don't have to go through this in class and we'll do an activity instead. So we're starting at the beginning. Just follow along with me. So know that mitosis, that the cell cycle, mitosis is just part of the cell cycle. So know that the main purpose for this is propagation of a lineage, which means you want to take your DNA as a parent and then pass it along to your children. And know that this is part of the cell cycle. This is the history. If cells had a reality show, this would be their reality show. The cell cycle is kind of like the microscopic version of the circle of life, just like the Lion King. And then highlight that number 1 that says cell division. We want to know that the parent cell, the cell cycle results in 2 genetically identical daughter cells. Offspring has the same DNA. That way all your liver cells look the same, skin cells look the same, heart cells look the same, and so on. And please know that number 2, maturation occurs after division. It means those cells grow up and they learn their job, just like you're going to do. They're going to learn how to be a liver cell, a skin cell, a kidney cell, things like that. Also know letter C. This is for normal growth and repair. In case you get some type of damage to any type of your cells have to repair those. The only ones that don't are your neurons and your brain cells. You kill those, you're in trouble. Please know under letter D that this requires DNA reproduction. This is called your copying your DNA. Remember that number 2, your genome, is your entire genetic makeup for your whole entire cell. If you look at letter B, just go ahead and highlight that in humans, that genome is 7 feet per cell. You got 7 feet of DNA in your cells, all your trillions of cells. That's an endless amount of DNA. No, just remember again, we've already talked about it, but we're going to go over it again. Chromatin. Those 2 phases DNA come in. Chromatin, loose state, bowl of spaghetti. Chromosomes, tightly coiled DNA. They call that the corkscrew noodle shape, just to give you a comparison. Know that the cell cycle happens in somatic cells. Those are normal body cells. So go ahead and highlight all of this. And know that the chromosomal content is 2N or diploid. Know that 2N, you got to get 2N from somewhere. So go ahead and write out somewhere the N plus N equals 2N. And go ahead and highlight that for humans this is 46 chromosomes. That's your 2N. So 2N equals 46. And if you divide to get what your normal N, half of that is, divide by 2, 46. Divided by 2 is 23. Ooh, so go ahead and highlight N equals 23 chromosomes from egg, and the other 23 is from sperm. Please know about the histones. Just those are proteins that help your DNA stay coiled up. And then sister chromatids, let's move on. Sister chromatids. We'll get to a picture. This is a sister chromatid. They're on the right. Go ahead and draw that. The easiest way to draw that is instead of, it says that it's an X-shaped molecule. Well, X's, they're not very easy to divide up. They actually look like 2 backward C's with a dot in the middle. That dot is a centromere. Please label as you draw. So, just know that this is half of a duplicated chromosome. And then you draw your sister chromatid out beside there. So if you look on the left, this is called a karyotype. That is I call it a chromosome selfie because it's all your chromosomes. It's all 46 of them. Pair 1 through pair 23. That will be very important whenever we come to a lesson later on. But just know, sister chromatids are connected to a centromere. The centromere is actually made of protein. So out beside centromere, put made of protein. That's very important. Alright, let's move on. Mitosis. Just know that this is ordinary cell division, and that the parent cell and daughter cells are exactly alike genetically. Whenever we come to meiosis, a process we'll learn later, the cell division for making gamete, sperm, and egg, they're not going to be genetically the same. So, let's look. So this is your cell cycle. If you want to draw this under the last page, not the very back of the last page, but that section under where it talks about cancer, if you want to draw that, this is a very helpful drawing. Instead of putting mitosis, you could put M. Most pictures put M there just to shorten it. So let's go through cell cycle part 2. These are parts of interphase right here. This picture helps you a whole bunch. If we look at, change color. If we look at G1, that's your first step of interphase. Please highlight that your cells spend 90% of the time in interphase. This is just normal hanging out, doing cellular processes, making proteins, shipping things in and out of the cell membrane. And interphase has 3 main parts. This is before mitosis. Interphase comes before mitosis. So know that G1, just know that it's ordinary everyday growth and activity and repair. And then know that there is a checkpoint here. That checkpoint, these checkpoints during the cell cycle are going to help prevent cancer later on. And then know that this checkpoint is called the point of no return. If a cell is not okay, maybe their DNA is messed up, maybe there's just been so much damage it can't function anymore, it's going to go into something, not G0, we'll get to that in a minute. It's going to go into something called apoptosis. So please make yourself that note. Apoptosis, which is program cell death. If your cells aren't okay, then there is a mechanism inside. It's a switch. It just says, hey, we're too damaged, we're hurting the body, and we need to take our cells out. And poof, cell gone. Alright, let's move on to S phase. Please just know that S phase, your DNA replicates. I call this the making copies. If you've ever seen SNL, and you've seen the skit where this guy stands at a coffee machine in the office, that's all he does all day is he drinks coffee and makes copies. And he stands around anybody that comes in. He makes fun of their name, like Steve Areno, Steve Rooney. And then somebody will ask him, what are you doing, Bill? And he goes, I'm just making copies. Making copies, man, that's my job. So, making copies. It's funny, but you'll remember it. G2, please know that this is where cells and organelles mainly enlarge and replicate. You're going to make 2 cells at the end of this. You want to make sure that you have all your DNA and all your cells. You want to make sure you have all your organelles, because otherwise, you can't function. Please know there's a second checkpoint here. And the question that cells ask themselves at the end of this checkpoint is, do we have everything we need for 2 cells? These 2 do. So that's G, this is interphase, G1, S, G2 in that order. Also, when you draw this, put your arrows in so you're not confused. So after you go through interphase, you go through your checkpoints, make sure you have anything, then you get to go through mitosis. So let's start with mitosis. Interphase, remember I would draw interphase, and then I would put out beside it my picture that I put 90% of time. This will go in the very back of your notes, that big old spot on the back of your notes. Please draw this picture, not this one. This is a microscopic cellular image, just to show you what it looks like under the microscope. Make sure you label everything, like the nuclear envelope, paracentrioles, chromatin. Remember that, spaghetti noodles. Plasma membrane, that's just cell membrane. You don't have to label nucleolus. Alright, so mitosis. This is nuclear division. Please highlight that. First you got to divide your DNA, then your cytoplasm. DNA is very precious. Got to take care of it first. So prophase. Just highlight what happens in each of these. They're right under there. I made them really short. So you highlight as I talk about it. So your nuclear envelope, your nuclear membrane. Sorry, it went backwards. Go back. Thank you. So prophase, this nuclear envelope is going to start to break down, and it's going to be rearranged into a spindle apparatus. Out beside that word, put the word spider web, because these look like spider webs. They look like spider webs that Spider-Man shoots out of his hand and swings from buildings. And know that this chromatin, that spaghetti noodles, are going to condense into those X-shaped, we call them backwards C with a dot, shaped molecules called sister chromatids. And these centrioles are going to move to the poles. Remember, that's like north and south pole on a globe. Out beside centriole, put Twizzler. They look like cut up pieces of Twizzler. So some terminology. Alright, let's move on to metaphase. So just go back and review what happens in each phase. We're going to go through hand motions. Break down a cell nuclear membrane. Turns to spider web, spindle apparatus. Centrioles are Twizzler. Let's go on to metaphase. Metaphase is really simple. Meta means middle. So highlight the M in meta, the M in middle. That's going to tell you where your chromosomes are. All that means is your chromosomes are going to line up on the metaphase plate, which is just a fancy word for middle of the cell. Make sure you label everything. Label your centrioles too, because they're going to be in every picture. If you look at your microscopic image down here, you're going to see centrioles right here, spindle apparatus, and then chromosomes line up in the middle. That spindle apparatus, go ahead and highlight. It's going to anchor to something called a kinetophore. It's the proteins, part of the proteins that are in this dot in the centromere. And those spindle apparatus is going to start pulling that sister chromatid apart. So you got DNA in both of your cells at the end of mitosis. So that's metaphase. Lines up in the middle. Anaphase. Go ahead and underline the A for away. Those sister chromatids are going to start moving away from each other. So remember, spindle. These are centrioles. Make sure you label that every time. These are sister chromatids. They've just been pulled apart. They're being pulled to the opposite ends of the poles, because we're fixing to go through and cut this little fella in half. So, know that they're pulled apart, and they're actually being walked to the end of the poles by these little guys called motor proteins. They pull those to the opposite end of the cell, because we're going to love to make two cells at the end. So if you look at this picture down here, that's what it's doing. These look like spirit fingers. So go ahead. I know that sounds weird, but it'll make sense later. These look like spirit fingers. So just put that out beside anaphase spirit fingers. That'll come in with our hand motions tomorrow. Alright, let's look at telophase and cytokinesis. These almost happen at the same time. Boom, one after the other. So telophase, some people like to say telo for telephone, because they say that this looks like a telephone down here. Looks like a 90s telephone. Maybe that's why I identify. So, just know that your nuclear envelope is going to start being rebuilt. That membrane around your nucleus. I'm going to erase that. It's going to start being rebuilt. And then your sister chromatids are going to decondense, which means they're going to turn back to those spaghetti noodles. And then there's something called a cleavage furrow. Make sure you label this later. To cleave means to split. And to furrow means to dig in. So if you want to write that down, cleave, split, furrow, dig in. You're digging in and splitting up this cell into two. Very important. And then cytokinesis. Know that this is division of the cytoplasm. Remember, that's that jelly stuff that holds all your organelles in place. And then know that cytoplasm and cell organelles are separated. Before, we've just been dealing with the nucleus. Now we finally get to look at your cytoplasm and your organelles.